My guest today is Dr. Gerry Quinn.
Yeah, I'm going to be talking about the spiritual healing sites as a fertile ground for finding new antibiotics. My main topic is Streptomyces, but I've also been looking into sites of spiritual healing. So Streptomyces, in case any of your listeners don't know, make more than 50% of antibiotics, but not just antibiotics, but anti-cancer compounds, antifungals, antivirals, and antiparasitics. Some of you might have heard of ivermectin, you know. So that comes from Streptomyces. Okay. Now, the reason we study Streptomyces, I mean, not just because it produces most of the world's antibiotics, is because we were coming to a shortage in antibiotics, and we had like a long period, maybe 30 years of great discovery of antibiotics, but they kept using the same techniques to discover the antibiotics. And therefore they came to this, replication problem where they kind of look in the same places and find the same antibiotics, like mostly around the world. So there's been a kind of shortage for 50 years. And, some people might say, well, there's plenty of antibiotics to go around, but most people have heard of antibiotic resistance developing in many countries, and maybe not so many have heard about antibiotic toxicity, from trying to revive old antibiotics that are like slightly toxic on the edge, but not, depending on the dosage. So a lot of researchers have been looking for new ways to find antibiotics, basically. And with the beginning of genomic sequencing, they began to find some of these antibiotic-producing organisms in extreme environments, like deserts or hypersaline lakes, or places where you didn't think that life would grow. And the theory was that if they have an adaptive physiology, then they'll be different from the normal bacteria that live in like a mesophilic field, and therefore they might produce more exotic antibiotics, and that's what people were looking for. And this is just a case of like sourcing antibiotics from a hop smoker in off the sea in Taiwan, where the Streptomyces are growing on the edge of the hot smoker, and they have an adaptive physiology which makes them produce novel antibiotics. And imaginatively, they called this antibiotic compound 1. It's not. I just noticed it now. After all the trouble, they came up with compound 1. But great. It was a new antibiotic. But also they can be found, in quite unusual places. This is a leaf cutter ant, and it's got Streptomyces growing on its back and its thorax. And supposedly, what the ecologist saying is that this ant uses the Streptomyces to farm its fungus garden, so it rubs its antenna on its back and then does some farming, and the Streptomyces keep some of the some of the other fungus away that might interrupt, because this ant farms fungus in a garden, so it's kind of unusual. So my co-workers were looking for antibiotics around the world, and the Gobi Desert and, the rainforest and, other exotic places. So, because I used to live in a place with quite a high pH, quite a high soil pH. Originally, I'm quite interested in plants, so I know that a lot of the plants around there are alkaline. So I thought, well, maybe there's some alkaline type Streptomyces there that we can have a look at. So I proceeded, and maybe in 2016, to head off around the hills looking for alkaline Streptomyces. But when I was doing this, some of the local people told me about this folk cure that involved some of the local soil. And then I was thinking, well, if I'm finding alkaline Streptomyces around the hills, then, obviously some of it might exist in this folk cure. But being a microbiologist or a biochemist, I was, when you start adding some traditional medicine or folk cures into your subject, I start thinking, how am I going to get this published? Because, the information I usually give is exact information. There can be nothing frilly at the edges, there can be no fluffiness. I have to give exact answers to, repetitions of experiments and stuff. So getting into I know folk medicine is very useful, for discovering new compounds, but on the edges of science, it gets a really bad rap, So anyway, I looked into this clay cure, and this old priest who died, he said that the soil that covers me will cure anything I was able to cure when I was alive, because he was one of the first priests to emerge after the penal laws in Ireland, which forbid priests, So, but he was in contact with, previously he was out in the fields, because he obviously couldn't have a church, and he was in contact with like the local folk healers, and he kind of knew knowledge, but more or less what he was saying was the soil is curative from the area, I think, not from his grave. But anyway, as is the tradition in Ireland, a tradition grew up around the soil that covered him. So they'd take some soil and they'd use it for various different cures and sore throats and all of that different things, So I brought some of the soil back to the lab. This is just the area it comes from, those cliffs are all kind of limestone, high pH areas, and it's quite a wet area, as you can see, because it's very green. Anyway, I investigated some of the soil and I found a new alkaline bacteria. I done some experiments over in Wales and also in Croatia with different clinical strains of pathogens, So first of all, you can see there's a diagram with a clear zone around some colonies, and that's how we screen things for antibiotic activity. We look like it goes back to Fleming. He looked for a zone on a plate, a clearing, and that was probably antibiotic activity. And below that's just a diagram. Streptomyces grows. It kind of grows like a mushroom circle. It has a nucleus, and then it keeps going out. Like looking for nutrients, because it's not a motile organism. It doesn't move. So its movement is making spores. And just keep expanding out. So this was a new organism, and we called it Streptomyces myelophorea. We genetically sequenced it. I noted that there was a lot of, I mean, I don't want to get too technical here, but antiporter genes. Antiporter genes are things that maintain a homeostasis. So if the ground around a microorganism is too acid or too alkaline, it tries to balance, the chemicals on one side with the other, so as it doesn't, so as it doesn't die from some kind of osmotic overbalance or something, Anyway, this particular organism, I don't know why, it had a really fragrant smell, like oil of wintergreen, like almost like an incense, and I thought that was it's quite fitting for an organism you find in a churchyard, but we'll come to that later. And then I tested the organism against, we got like 36 clinical isolates from a hospital in Croatia, and we tested them against this particular organism. And they were all multi-resistant. They all had resistant to this, to that. So, and it killed. So the WHO make a list of antibiotic resistance organisms that they should really get a grip on and have new antibiotics for. So we ticked the box on 3 of them. What the first one was methicillin resistant Staph aureus. Most people know that, MRSA. And then we had the vancomycin. Vancomycin's a step up in antibiotics, so it was able to kill vancomycin resistant enterococcus. And then one of the newer antibiotics, carbapenem resistant Acinetobacter baumannii, it was able to defeat that, so that was quite a good haul. I mean, it didn't kill absolutely everything, but it was quite, And then I went on to publish some more papers, because I searched the hills again for more samples of, Streptomyces, and we went on to find like antifungal Streptomyces, antibiotic, antiviral. I was starting a project on antiviral Streptomyces that kind of stopped all of a sudden, but it did have effects against SARS, COVID-2, and then I was doing some anti-cancerous, so some of the molecules produced by Streptomyces, they cover a couple of functions at once. They can be antiviral, they can be anti-parasitic at the same time, or antibiotic, antifungal, antiviral at the same time as well, Okay. So that research was like maybe 6 years ago, 7 years ago. And during the time I was researching, I began to collect motifs, and I realized that in the first publication, I was strictly biochemical, just discussing microbiology, and I really didn't give much space for the folk cure aspect, Well, it's not I ignored it. I said it came from a folk cure, but really I didn't mention anybody praying, and I didn't mention any of that because I thought as soon as I mention this, my likelihood of getting published might, and people might not take it as seriously as soon. But the more I looked into the new discoveries in Streptomyces and thought about, what the folk cure people were saying, I was like, it's like this thing, scientists discover water is wet. Some people knew already, but they didn't know exactly. They couldn't chemically and physically describe it. So that's what I was thinking about people that done. I was like, why do they do this, and what does that mean? So first of all, I assembled all the the geochemical characteristics of Streptomyces discovery and sites of spiritual healing, and some of the main motifs, although not exclusively all, were karst environments, which is limestone environments, high pH, places with lots of caves, poor soils, very thin, weak soils that are really not good for farming, and obviously a long history of folk medicine somewhere in the background, and some people might recognize these names, and it's not my intention to denigrate anybody's spiritual beliefs. It's not my intention to say, aha, this is the real meaning. This is just some observations I've been making. So, places like Lourdes in France is all limestone. The valley's all limestone, and the caves are all limestone. And it has the typical qualities that you would find a lot of Streptomyces in, okay? And that is the same for, Fatima, places of pilgrimage, Croatia, Medjugorje in Croatia, Bom Jesus de Lapa in Brazil, okay? And there's another one in New Mexico, which is slightly closer to you, Tom. It's a Sanctuary de Chimayo in New Mexico. But that's not limestone. That's built on a hot spring, which is an extreme environment. And hot springs are, a place where you find Streptomyces as well, So anyway, but, you could say, well, maybe coincidence, but there are even older traditions. So there's a place in Canada where they use glacial clay, and it's used by the Helsier Indians, and that's been thoroughly examined in some labs in Canada by, I think, Julian Davies. He's dead. He was like the father of antibiotic resistance, exploration, and they found that this clay killed lots and lots of pathogens. Similar, there's a folk cure in Jordan. It's the red clay they use to rub on their skin. And more recently, they found this ancient cave in Apullo and in Italy, which is this is a illustration of the dancing wizard or the dancing god, where there's lots of Neolithic markings, and they've discovered like some new strains of Streptomyces, so it's not that unusual. So one of the other motifs is, my theory is the Streptomyces is found in some spiritual it's found in some places, and then the local people notice, and they describe it as a spiritual, as a spiritual place, holy soil, holy water. And something must have happened, and somebody got better, and then they're like, well, that's a God-given gift, and that's my kind of theory, and this is traced through many religions over time. So the place in Bow, which I originally found the Streptomyces, it was built on top of a Druid amphitheater, and previous to this, there's some stones 400 yards away. They're Neolithic. They're like 4,000 years old, and they have like ceremonial markings. The cup and ring, and they've been so it's like ceremonial stones. So I don't even know what kind of religion it was, or what kind of ceremony, but it's just an observation, that we made, and I'm connected with it, like a good scientist. I'm connecting it. So even places like St. Catherine's Monastery, which is a Christian site at the bottom of Mount Sinai. Obviously, Mount Sinai is associated with the Jewish faith and the bringing of the Ten Commandments to Moses, But even before that, there was the Bedouin tribes and different tribes that revered, that part of the mountain, and there was some there's some association with like solar deities or solar worship, from that area, So it's not. So basically, the cultures build on top of each other. So that kind of sorts out, where do I locate Streptomyces? Maybe there's new areas to look in. But the reason we've reached a crisis in antibiotic production is obviously antibiotic resistance and no new antibiotics. So if, say, we do find a new antibiotic, are we just recreating the cycle? So in 30 years, the antibiotic resistance, should we not be kind of trying to look where antibiotics come from and look at their role in the ecology of the chemical ecology and say, well, how can we design a better system that won't become resistant? So and that's where looking at the spiritual motifs behind and the cult traditions. But this is it's more fluffy, it's more, I'll say that at the beginning, it's more fluffy than scientific, but there's so many unanswered things. But as a researcher, if I don't have unanswered questions, then if they're all answered, then I'm kind of out of a job. So it's a real puzzler. There's not so many experiments can be done, and it's harder to trace those old people who might know what happened. And when I actually do talk to some old people, they have different versions of what they think happened. So okay, so just diving deep into this spiritual metaphors, about holy soil and stuff. So holy actually comes from an old English word meaning like whole, the whole. So we're kind of reductionist. We're looking at a part, the antibiotic that we pulled out. So it's good for microbiologists to remember that all of these molecules evolved together in a soup. They didn't just, antibiotics arrived on their own. They were all surrounded by other different molecules. I've just kind of put in vertical commas, no antibiotic is an island unto itself. They're all part of the one milieu. So but when antibiotics are produced, they don't just produce one antibiotic, they produce many other molecules, and these have been they've just been ignored. The race to purification is mostly what pharmaceutical companies think about, and so when it comes to time for the organism to produce the antibiotic, it produces another 100 compounds, but traditionally they've been ignored, for the last 70 years. But the research has just actually started into, what are these other compounds, and do they trigger any effect, because, we might be looking also for antibiotics and holy water or holy cures, but it might not be the antibiotic that's triggering any effect. You know, you have lots and lots of different compounds, and you have a whole microbiome in your body that might. So some people done some research on Microbacterium vaccae, which is found in soil, and they were wondering why gardeners were happier, you know. And then they traced this biochemical pathway and found that it was involved in the release of serotonin, you know. So it interfaces with the immune system of the microbiome in the stomach, and it sends a message to the brain to release serotonin. And I was like, oh wow, you know, I mean, so Streptomyces group is kind of related to these Microbacterium group, you know. So as I was saying, the idea that they use one compound to treat a disease, it doesn't happen in nature. Nature, although it looks like the antibiotic is the only thing fighting its competitors, that's not true. But there's many, many other compounds, you know, as I said, hundreds, and these can help the antibiotic to be stronger. It can help it to penetrate the whatever it has to tissues more effectively. And that obviously means that you can use lower doses of the antibiotic. And it can also help with the toxicity, you know, of some antibiotics. So things like biosurfactants, people don't know biosurfactants as things like soaps and stuff, but that increases the permeability of some cells and helps the antibiotic to penetrate through, or antioxidants, which will keep the antibiotic fresher. Or some new discoveries of iron scavenging compounds called siderophores, and they've been incorporated into some new designs of antibiotics, so that maybe the pathogen thinks, you know, it wants to scavenge some iron, you know, and if it's attached to a siderophore, the organism brings it back into it's like a Trojan horse. It'll bring the antibiotic, which is conjugated to the siderophore, back into its cell, and that kind of helps more with gram-negative infections. Gram-negative is just a type of bacteria that causes more trouble than gram-positive. So, and even the pigments that people think are useless are helpful in increasing the activity of the antibiotic. So, you know, now they have incorporated some of these adjuvants into some new therapies, you know, but it's kind of early days. But they weren't always. Hold on, this is kind of a diagram. I don't know if you can see the top of that. You know, so the first one up the top where it just says antibiotic is the traditional way they made antibiotics, and the one down the bottom is something they're moving into. It's kind of a combination of compounds. It's more, I don't like using the word holistic, only because it gets a bad reputation, but it is it's more considering the context of what happens in nature. You know, it's a contextual approach rather than, you know, just a one fits all approach, you know. But what I've discovered recently is if you use 2 or 3 compounds with the antibiotic, that stops the emergence of antimicrobial resistance happening as fast as it would with one compound, you know. So for an organism to get a handle on one compound and say, okay, so we're going to find our way around this, but if you add 2 or 3 other compounds and they're not necessarily antibiotics, then it takes a long time before any resistance develops, you know. So and then it's closer to the natural situation. Okay, so back to the airy-fairy stuff. But, you know, originally, you know, in certain parts of the Bible, you know, you'll read Jesus cures somebody and he says, go away and sin no more, you know. And you're like, well, how did he cure them? No, what's the cure, you know? So a lot of these traditional practices emphasize the mind and the body and the spirit, you know, as a whole. And you'll say, well, yeah, I mean, that's all that airy-fairy stuff. Just give me the antibiotic, you know. But most of these cures involve prayers. They involve a ritual. And often they involve silences. The bow cure involves praying for somebody else's intentions. And then you're meant to not say anything until you get home. Something like that, you know. But recently they discovered that this positive thinking, this focused thinking, has an influence on the hypothalamic pituitary adrenal axis, which influences immunity. So that makes the immunity even stronger. So when Fleming discovered penicillin, he and experimented on it. Obviously, he says, well, without a good immunity, is is no good, because you have to have a working immunity, for the penicillin to work. And the other thing about the folk cures is that, I mean, they're all miracles, because they don't know how they work. They have no clue. It's beyond the medical is beyond the thinking of what is normal, And I mean, that's where researchers try to go. They say, well, we know up to this point, what do we know beyond that? Well, we have to do some research, and we have to do some experiments and do this and do that. So, Streptomyces, as I was saying, I keep banging on about it, but it's been under the microscope for 80 years, and they use it to make tons of antibiotic. Okay, but in the last, so they've developed a whole way of thinking about Streptomyces, and it's not that they've closed the book, but they kind of say, well, we know everything about it, but actually they don't. That was part of the problem, and creating new antibiotics. So in the last 20 years, what they discovered is some of the species of Streptomyces they thought produced 1 or 2 antibiotics. In its genome, it encodes 10 times as many. So if it makes 2, the genome encodes for 20 different antibiotics, and these antibiotics are described as silent metabolites, or cryptic. They call them cryptic metabolites. So this is one of the mysteries of Streptomyces researchers, is how to stimulate the production, and that's what it interfaces with, like the folk medicine, because folk healers they have a day, and they have a time, and they have a cycle of the moon or something, or a plant that lives beside these Streptomyces, and that's the time to get them, So even when I bring a Streptomyces out from the outside into the lab, and I try to recreate the conditions in a lab, it's not, half the time it's not successful, because we don't know what's amiss. We have all these really complex growth materials, but sometimes it's, we take a bit of the soil, and say, okay, maybe that's not even enough. It might be a particular temperature in the year, or some other atmospheric things, sunshine or stuff, So, and so they thought they had Streptomyces figured out, okay, and then, like, I think it's 5 years ago now, they discovered. So you get planktonic bacteria. They're all individual, and you get bacteria that grow in colonies in a plate. And then, like, some new researchers in Canada discovered they put a stone in the middle of some agar and discovered that the Streptomyces stretched. So they discovered a new stretch morphology, But that's like 80 years into the research, and you're thinking, well, why didn't they know about this before? But yeah, they didn't, They just put maybe it was an accident, I don't know. They put a rock with some minerals, and then they observed a stretch. And what? So why does that make any difference? They've got a new morphology, and the difference is that each new morphology does something to the control of the antibiotic production. So if you fit the Streptomyces into a tight space, it produces something. And then if you have it in a liquid, it produces something different, And if you have it at one temperature, it produces something. And then if you have a very low temperature, it produces something different, So they're like prima donnas of the bacterial world, And they also discovered biofilms, which they thought, I used to work on biofilms, which they thought would have been a natural, but they're like, oh no, we don't know. And then all of a sudden, because biofilms are a type of bacterial morphology where all the bacteria join as one organism. They have central control on this biofilm, this blob, which usually the control is with each bacteria. So, and that produces something different. So. That kind of interfaces with the folk healers, because they have pattern days. When is the best day to get this cure? What is like usually like the 3rd of March or the 5th of August is, it depends which country it's in as well for the temperatures and the seasons and stuff, you know. So anyway, the last thing I'll say, so there's many, many unknowns that I really want to investigate. You know, there's a lot of, I've written up there tree rags, actually. You know, beside a lot of these holy cures, they hang rags on trees, or, you know, different traditions. The Buddhists have them. Some of the American Indians have these bits of cloth hanging, you know, and in Ireland it's quite a popular thing to hang rags on the tree. And I've been trying to figure out, you know, I know I was reading some research recently about Streptomyces in clouds. You know, some person was gathering Streptomyces in clouds, but he was just seeing how they're transported in the air, you know. But I'm interested in why people hung rags, you know, and cotton rags, you know. Radioactivity is a big one, you know. You might not think, but limestone obviously gives off radon gas. And the limestone I've been exploring is about 300 million years. So they've had the evolutionary pressure to do something, you know. I've been playing around with Streptomyces in the lab, zapping them in different ways, and we haven't got the result yet. But, you know, I'm interested in what's been. I can't throw a lot of radioactivity around the lab anyway, but I can throw microwaves and UV radiation and other kind of stuff around, you know. I mentioned at the very beginning of the talk the smell. We can smell Streptomyces volatiles better than a shark. You know this myth about a shark smelling blood in the ocean? We can smell Streptomyces better than the shark smelling blood. And parts per trillion. No, sorry, parts per billion, not trillion. Parts per billion, not parts per million. We can smell, it's like eight parts per billion. And why? You know, why? I want to know why. Why are we tuned into that particular smell? So anyway, and also, you know, we're not so far along with the genomic sequencing, but, you know, there's lots of different aspects to the cures. As I said, it was antifungal, antiviral, you know, but there's a couple of other ones that the folk people say there's cures for infertility, but we haven't kind of linked up the research for that as yet. So I'm quite interested. There's lots of ways to go and directions in my research, you know. So I think that's about it. Sorry, there's not the end slide, you know. That's the end slide. Oh, sorry, if people want to, if they can read that, if people want to read about the research, I'll just put it that they can freeze frame and go to that link. Is something I wrote, my latest thing in the Journal of Religion and Health, you know. So that's it, Tom.
Oh, okay. Incredibly interesting. I'm curious as to how alone are you in doing the research that you're doing? Are there lots of other people that are looking into this stuff and testing, etc.?
The Julian Davies in Canada, who's dead. Sorry. He was 94, 95. So no, I definitely the spiritual thing. No, I don't know anybody. The folk medicine, I know one or two people. Oh, they do Streptomyces, you know. The spiritual thing, no, I don't know anybody.
Okay, I'm curious. I heard elsewhere that there may be 38 trillion microbes living in the average human body. And do you think we understand what's going on with them and which ones are beneficial and how they interact with each other and all that? And if we take antibiotics and we kill off the wrong ones, could we be doing some harm?
Yeah, and antibiotics is as I said, it should be contextual. You know, we should know I mean, it was an emergency. We'll say that it was an emergency. But, you know, I mean, there was a lot of deaths back in 1940s, and I appreciate, you know, why they did what they did in the 40s. But I think they should really be looking about, you know, I used to work in a pathology lab. My boss never touched antibiotics. And it's not that I wouldn't take an antibiotic if I had a really bad, you know, septic infection. Obviously I would, you know, but we should be looking closer at them. Yeah, and what they do. I mean, a lot of antibiotics work on the premise of controlling protein production. They go right into the ribosome, you know, and stop it. A bacterial ribosome, which is different from a human one. And that's the reason we can take antibiotics, because supposedly they only work on the bacterial equivalent. But we should definitely be looking at the ecology. There's a lot of people investigating the microbiome just now. We might be able to do some good things if we are proportionate and mimic what's happening. I think part of the problem of antibiotic resistance isn't that we're using antibiotics. Antibiotics have been around for hundreds of millions of years, and so has antibiotic resistance been around for hundreds of millions of years. What we are doing is we're unbalancing the chemical ecology. We're making massive amounts of one particular thing, and it may have the butterfly wing effect. It may have effects on the other end, and we don't know. So no, we should be cautious. We should use antibiotics very, very cautiously. And definitely not. We should be cautious about anybody using them long term, giving them to animals. And we think we take a lot of antibiotic, but they give animals tons, for the farming industry and stuff. And I mean, is that really necessary? Is it really necessary? So I mean, I think antibiotics are a gift from the creator, or whatever you want to call it. It's a gift from nature. So really, we discovered a really interesting thing. But we're in 2025. We should have scientists that can look all through that and say, what's the chemical ecology? What we unbalancing? So microbiome is a big thing. There's not enough people involved in the microbiome projects. I mean, the lung has a microbiome, the brain is a microbiome, Wow. I wouldn't even think that. Amazing, So I was looking into, there's organisms like Streptomyces that exist in the body. I was looking at maybe some of their metabolites are sending signals around the body, doing, you get some of the microbiome actually produces antibiotics, the normal microflora produces antibiotics as well. My first subject used to be in a lab in Florida. I was looking at what antibiotics the skin produces, with some, and some of them they were using for, they were modifying them a bit and using them against HIV and hospital super infections. So that was really interesting, So we definitely don't know enough,
What do you think, just as an aside, the whole issue of overuse maybe of hand sanitizer? Do you think you're killing stuff on your hands that's supposed to be there, or what do you think about that?
Yeah, that's germ theory stuff. I mean, it has its function maybe, but you're just breeding the most resistant organisms by wiping off the easiest to kill ones, If in a healthy environment, the soil is full of useful microorganisms. It's not all full of anthrax, So the hand sanitizer is definitely overdone. You're killing all your microflora. The microflora help your body, It's not that you should be completely dirty and smelly, but you're killing a lot of your helpful micro, and we don't fully understand the extent to which they help your health.
And then maybe if you're a parent of young kids, maybe you can overdo the keeping everything super sanitary and letting the kid roll around in the grass a little bit. Might be good.
That's a definite, I think that's a given, And also at that age, you should be, unless you're kind of compromised, you should be really bumping into things that you can defend your skin cell. I've done this in Florida. Your skin cell can actually tell a nasty bacteria from a good bacteria, just to put it in simple terms, and it will make the appropriate response with your innate immunity. That was just to be my subject. It kind of still is, and release natural antibiotics from your skin. And if you keep wiping it down with alcohol, all the balance is gone. I mean, wash your hands. Yeah, wash your hands, but keeping wiping alcohol and stuff is just, it's a bit too much for a normal domestic kind of use.
Do you think there's a role?
Wash your hands before food.
Do you think there's a role in sunlight on the biome on your skin, or what do you think in general about sunlight?
Yeah, I was reading some research on that the other day. Is completely, sunshine even does things to your microbiome. I was reading research that the sunshine penetrates through some of your layers of clothing, and right in that. I can't remember. It was like last week I was reading this paper and I was like, that's amazing stuff, So sunshine is completely beneficial. Maybe not slathering your body with sun blocker, but you hide underneath a tree if you get too much sun. That's what I used to do in Florida. Hide between the trees. No, we did. It's just we're just beginning to look at the microbiome, and to interfere too much in the processes that we don't understand. I'm not saying we should live like Stone Age people, but when I first saw the hand sanitizer making appearance in 2010 or 2012, whenever we had that flu, the last big flu crisis, I was like, somebody's making money. It's completely over the top. They're just like throwing it out in the community, just like wash your hands if you want to be like a bit clean. But that whole special antibiotic, that antiseptic, thing is, yeah, I'm suspicious. I'm suspicious of the motivation that might not be health.
So I'm curious about the discovery of ivermectin. I was just looking it up. I think it was 1973, maybe. They found it near a golf course. Was anybody using that particular soil for healing before that, as far as we know? Or that was just like a brand new...
on the golf course?
Yeah.
No, I was looking into that as well. The Tama golf course is beside Tama graveyard in Japan. Because I was looking into all the occurrences of Streptomyces in graveyards. And I know people don't want to know that you find them in graveyards, but, they degrade things, Tama graveyard beside Tama golf course is notable for the discovery of it's like 4 or 5 species of new Streptomyces making new antibiotics. So, I mean, Streptomyces is found all over the place, but maybe people like sampling graveyards, but I'm really surprised at the amount of times people are sampling graveyards and find Streptomyces with new antibiotics. I'm like, that's a bit strange, but yeah, I was looking into it. But Tama golf course and the graveyard was built, it was like 1910s or something, So I don't know. The Japanese have some kind of system of like ancestor worship and reverence and of all the soil in the graveyard and stuff, so I'm not so sure that anyone tried the soil previous to that. I can't get beyond the first construction, Google one or whatever search engine I use doesn't find that, But
So
definitely I want people to go out and swab parts of their favorite healing place and see if there's any Streptomyces, definitely,
I mean, can ordinary people do that, or they would have to send it in somewhere to get this done?
Send it in somewhere.
Yeah.
Actually, I'm working with somebody that's tried a homegrown agar. I mean, you can buy agar sold in the Chinese food shop. All you need to do is make some very weak agar, maybe with some bit of soil, and then But the Streptomyces is a bit like growing orchids. Sometimes we add things to the agar to stop the overgrowth. Other organisms will grow way over the Streptomyces to begin with. Although it produces antibiotics, it takes a while to get off the ground, so you'll get a plate of fungus instead, or, so, but some people have done it in school projects. Yeah.
I mean, what would be the logistics? Like, say, if I thought that there was some soil that I wanted you to test, how hard would that be for you to get that in your lab and take a look at it?
Not very hard. Test the right soil.
Okay.
I would say slightly above pH 7. Just take a swab, put it back in the swab holder and post it, because it's not growing anything at that stage. So you're not sending microorganisms, you're just sending a dirty swab, so. Yeah.
Is the process described anywhere on a website where an ordinary person could read it and just follow the steps and send you stuff, or?
Yes.
Okay.
Yes. Many sites, but it's not for me. But some people have been sending me stuff because they've explicitly sent me an email and said, "Can I send you stuff to look at from this holy well. I've been doing a lot of holy wells recently, and I said yes. So I'm doing, sorry, blah, 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 Jerry. I'm doing these 3 holy wells that are named after specific diseases, and we've discovered like lots of different streptomyces in them, and I'm trying to link it up to what the diseases are. So yeah, people do it all the time, you know.
So I probably missed the timeline, but did you say that maybe for thousands of years people have been using healing waters and healing clay? How far back does this go, do you think?
The stuff in Canada was meant to go to Neolithic times, you know. Given the ceremonial stones in Bow, if some soil ceremony was the markings are at least 4,000, you know, years old. I would think, you know, I mean, there always has been this like fertile soil, good soil, bad soil, you know, but I'm pretty sure there was a healing soil thing going around as well, you know, and healing waters. I'm pretty sure. But the furthest I can trace it back is, yeah, a couple of thousand years. But then supposedly, you know, civilization is only 6,000 years old. So, you know, some people say that, you know, so.
So I'm curious, the people who are using these a couple thousand years ago, like if they found healing water, would they just bathe in it or put it on their skin, or would they drink it, or would they process it somehow? And the same with the clay, would they process that? Or
there's some people, Nubians, so whatever, a place in Africa or something, some Nubians make this beer, okay, and it has streptomyces in it. So they dated some ancient bones and discovered tetracycline. Tetracycline is an antibiotic for streptomyces, widely used. And but the thing about tetracycline is it stays in bones, and they discovered it in their teeth. They studied tetracycline in their teeth from these ancient Nubian bones, you know. So I think that was a couple of thousand years old, you know. But they done some mass spectrometry on it and says, oh, what's this? You know, and it was tetracycline. And then they looked at their folk practices. So they didn't, and this traditional beer they brew grows a type of streptomyces, you know. And they're saying, oh, well, maybe beer is, you know, I mean, everybody drinks beer. It's not because you're ill that you drink beer. So, you know, but the archaeological investigations into a lot of this stuff hasn't been happening a lot, you know. So we know, oh, sorry, the ancient Egyptians also used to use some cures that might have involved some streptomyces. There's some ancient papyrus. I've written it down somewhere. They might have used some of these as well, but you know, it's fragmentary, you know. Some of that's in the article, which I can't remember the specifics of some of it. So,
but
it's in a newer article, actually, not in the original ones.
But again, currently in Western medicine, they think this is all superstition, and they're just not open to this at all about what they did 2,000 years ago to try to heal people. Is that true, or is the door open a little bit to looking?
There's
back?
a crap. They are adopting combinatorial approaches, like add 2 compounds or 3, as a really new thing, you know, which is harder to test than one. But the other stuff I'm writing out, no, it's like some of that's just fresh off the top of my. I mean, obviously, I'm digging into research there, you know, about the positive mindset and hypothalamus axis and stuff. That's kind of established research, but they don't look at that's from another type of research. That's not from taking folk cures, you know. That's from another positive reinforcement thing, you know. It's not from folk cures. So nobody, I don't really know anybody that's kind of looking at it from the same perspective as me. But then I grew up with folk medicines, so, you know, I mean, I grew up knowing about them. I didn't use them, but I grew up when I was younger, I was completely fascinated. I thought that the people that dispensed folk medicine were wizards that I had read in my schoolboy books. And I used to badger this one particular person because although the cure I was examining here is well known, Most of the other cures in Ireland are a secret, and it's passed down from father to son or father to daughter or something, and they have to keep the secret. They have to give you the medicine when you ask, okay? They can't take payment. They can't doctor it. They have to keep it as religiously as pure as what they were told, you know? So I was fascinated with this process, and I had some relative that lived up the mountain, and he had the cure. And I was, as a young boy, I was always trying to get it off of, you know? But obviously, he just passes it on to his daughter. He passed it on to his daughter, you know? And by the time I got to his, to ask his daughter, she was like 70 something. She'd forgotten like what he told her. You know, she'd moved into the city a long time ago, and you know, moved around the world. So yeah, I was fascinated when I was young, and it came back to me when I was researching Streptomyces about all these, you know, all these folk cures. You know, so the reason nobody else is on the path is because maybe I've had a different journey through what I was doing, you know, and we all don't have a similar journey, you know. I've done many occupations, you know, so it's kind of different things fascinate me, you know. So,
but if you actually had something that could help human suffering, why would you keep that a secret? I think it would be great to write it down and share it.
Why would you keep it a secret? Because none of them could write in those days. Really, 100 years ago in Ireland, like only the educated classes wrote, you know, so they pass it down orally. But the secret was because I think because they had to keep it faithfully, like exactly, you know, it's a secret to them, and they're got some solemn oath not to adulterate it, not to use chicanery, and not to muck around with it, because that's the gift they've been given, you know. So they keep the gift, and they don't, you know. So it's not that they won't tell anybody else. I mean, it is a secret, but they don't want it diluted, you know. They don't. That's what actually happened in ancient Egypt. They diversified to such an extent that, you know, they had subdivisions of subdivisions, and each person, you know, because they were making money out of it as well. And each person had a subdivision of, you know, brain, whatever they treat in brains, and whenever they treat in digestive conditions and stuff, you know. So that's kind of what happened to the Irish cures. They all have like a treatment for a wart on your finger or something, you know, and it's, but I think they're trying to keep, in my opinion, they're trying to keep it faithfully, you know.
Okay. And then some of these cures are quite complicated, right? You don't just take this clay and put it on your skin. You have to get it from a certain place at a certain time and process it in a certain way or something, right?
Yes. They're called pattern days. At a certain time of the year is in maximum effect, you know. So I think that's maybe why, you know, some people like lures or something, they say, oh, well, let's test the water. Is it an antibiotic? But maybe they're looking at the wrong time, or they're looking in the water. Maybe they should look in the stone and see if they can pull out any Streptomyces. But Streptomyces won't grow in one day like normal bacteria. You have to cultivate it quite carefully. It's like growing orchids, you know. And so not everybody is a Streptomyces cultivator, you know. So the people testing might be chemical testers and saying, yeah, I don't see anything. What are we talking about, you know? And the effect, as I said, might not be antibiotic. It might be some antigen from the Streptomyces that stimulates the microflora on your body, and it produces a subreaction like the mycobacteria vaccae, you know, given the beneficial effect from gardening, you know, that releases serotonin, something like that.
Okay. And again, if this whole big pharma thing, if they're already selling drugs, making a lot of money, they could easily set up a test saying, look at this, doesn't work. This folk cure doesn't work. They could set the test up very easily to fail, right?
Yeah, I mean, it seems, I don't know, Tom, but it seems to be a bigger thing that a lot of folky type cures are, a manuscript appears saying, yeah, it's no good and it's probably toxic, you know, even though the good thing about, you know, folk cures is they've been field tested for thousands of years, They've been on the go, for quite a long time. But I do see I do see some manuscripts, and I'm like, why did they write that? Like, what was it? Lavender oil, And I was like, no, that looks innocuous. And they're saying, no, it's harmful, And I'm like, oh, yeah, they've been using lavender for a long time. How did you figure that one out? And I was like, oh, who's this sponsored by? You know? But yeah, innocuous things like that. Like people say, oh, okay, things have been used for thousands of years, and just mysteriously papers appearing saying, yeah, it might not be helpful to use that. No, it might not be good. So I'm skeptical about their motivation for publishing such articles,
So the whole mind-body thing, it is tough to measure, right? But you said that there are some studies, big studies, where they are measuring some mind-body stuff. There's some research going on there, you just said, right?
Well, no, definitely. The hypothalamic pituitary axis. Yeah, definitely. Adrenal axis, sorry. Yeah, they look at a lot of people look at the effects on the microbiome and the gut. I mean, that's the first place to go. I mean, there's obviously microbiomes all over your body, like the skin and different places, but they look to see what kind of measure signals between, the gut, the second brain, and the main brain.
Okay.
So well, I mean, the brain and the gut are they're derived from the same tissue, morphologically in the development of human being. The brain and the gut and the skin derive from the same tissue, So they're all neurologically active, Starting off research, I used to do frog research,
Hmm.
And frogs produce a lot of neurotransmitters on their skin, but also in their gut and their brain, so it's all kind of linked,
Do you have any thoughts on grounding, on walking barefoot on grass? Is there any health effect whatsoever of that?
Oh, so you're going completely tangential on me, right? I do barefoot running, Tom. So
Okay.
I love going to nice green fields on Ireland, and then running on the grass is excellent. I can feel the warmth of the moisture on the grass. I know it's summer because I run sometimes barefoot in winter, but in Ireland it's not so much snow. But it's great. I love it. Grendon, no, I do. I was thinking about, because they used to send people to caves as a kind of hydrotherapy. I mean, there is something to do. It's not another area that's not still studied enough is how much, it's like, what is it? Plastics give out like positive ions, and rocks, mainly have negative ions, so you're trying to, and your cells are like, what, -7 millivolts or something, so your cells are kind of negative, and the theory of, I'm going tangential on you, Tom. The theory of antibiotic peptides uses the charge on the membrane of bacteria being different from the humans, so the peptides are what they positive, and they break the negative of the bacteria. They break into the negative charge on the bacterial membrane. So there is a lot of study to be done on the proper balance, of positive and negative. But I really don't know how to appreciate it, because a lot of substances we walk on and walk with are synthetic. I started to wear these barefoot running shoes because there's lots of thorns sometimes in the grass with farmers sawing their hedges. They find a nice field and make sure there's not a thorny hedge beside it, But I like the theory of grounding, but it's really not. Also, the research is just, if you can't make a living with the research, nobody's going to do it. A lot of this folk cure stuff, it's my hobby. Nobody pays me for doing it. It's self-funded, and through associates and other generous university people that give me bench space and chemicals, then I can do some research on it. when I publish a paper, then nobody pays me. I pay the publisher to publish the work, you know. So it's not, I do other stuff to get money for science, but the antibiotic research from natural sources is unfunded. I mean, I fund it, you know. And also in the very beginning, I'm you know, I was like, this is not, it's not, none of the organisms are patentable. Okay, they're natural organisms, and the cure has been around for thousands of years. So it's not mine, it's somebody else's. So I wasn't, in the beginning, I was like, I'm not jumping into somebody else's thing they've had free for thousands of years, because the Irish cures are free. You know, even though they're a secret, the person that has them has to give you it for free, and is not allowed to take any gifts, although people try and give you a gift, you know. Is not allowed to take any money for it, you know. So me jumping into that, and I mean, I don't live on fresh air either, you know. But no. It's all self-funded, you know.
So is genetic sequencing and or AI, are they big parts of what you're doing now, or advances in those areas? Is that helping?
Genetic sequencing helps, okay, because it can compare it to an antibiotic producing organism and save time, and I can look for patterns. But some of the antibiotics are, they have a gene cluster, but some of the gene cluster makes an enzyme, which is an antibiotic producing enzyme. It's a huge multi-enzyme, okay? But some of them mix and match. They plug and play and make a variety of different things. So it's not going to tell me exactly if it's a new one, you know. But I'm doing, for this Holy Well project I'm doing just now, we've sequenced some of the strains, and like, oh, that's really interesting. But what I'm interested in is the genetic sequences tell you if it's similar to something that has been found before. But I'm more interested in stuff that's not been found before. So there's the stuff that has no pattern recognition, because that's more interesting for something that's more resistant to antibiotic resistance, you know. So you don't want to find something that organisms have already become resistant to. You want to find that the gold standard for finding new antibiotics is find a new template molecule. You know, so a lot of antibiotics are built on similar templates, but to have a really, really efficient one, you want to have a new template in the middle, you know. And that's what I'm looking for. So it does help me a bit to produce manuscripts, and I say, it's like this and it's like this, but the real interest is finding something that hasn't been found before, you know. The AI, AI is like plus and minus. It's very early stages. But I thought, AI is so clever. Wow, it can find all those documents for me, you know. But I can Google it just as fast. And also it tells me some lies, unfortunately, you know. I was like, okay. And I typed in the exact same question from my manuscript. Okay, is there any discoveries of Streptomyces at holy sites, blah, blah, blah. And it threw me a list, and I'm like, that's incredible. Somebody's done that. And then I'd nearly written the manuscript, and I said, right, I need to double check this just, you know, for references and stuff. And then, so I'd put it into Grok or whatever again, and then say, no, there is no evidence. And I'm like, you told me, you know, and it says, no, it's been found in a similar area. And I'm like, oh God, no, the editor will just shoot that down, you know, because I had 5 or 6 things lined up. I'm like, well, that's wrong. And then I've also asked Grok about my own research. Obviously it knows less than me, and it knows less about my publications than me, but the publications are out there. So yeah, it's good for assembling data and getting in, but it also replies in a certain way. You know, let's break it down into this, and this, and that's not always the way I think. It's good for organizing your thoughts, but yeah, I trusted it too much. So now I have a level of trust in it, and I'm like, that's enough just now for what you're capable of. Not that I think I'm more intelligent than the AI, but, you know, I'm like, you know, did you actually look at this research paper that you didn't find, you know, something like that.
Final question, I think. I don't know if you want to share any opinions on what is happening with the WHO, what they're doing, or what they could do better.
Yeah, and I think they can do better with getting some independent science. I think there's too many. I want people to do better. I want health around the world, but there's too many. I don't want to share too much, but in my youth, I was protesting about this and that and the next thing. And one of the cleverest tricks in recent times is people that maybe not people that don't really wish well upon the world have taken some of these organizations and they're using their heartfelt concern about humanity and they've twisted it around as a tool, and I'm like, that's a trick. That's a real confidence trick. They've taken people's concern about health or whatever they're talking about, or I used to be quite concerned about, and I still am concerned about the environment. They've taken it and they've pivoted it, and they're saying now you're going to work for us, but you think you're still doing the same thing, So I think the WHO has gone the same way. I used to have a tremendous amount of respect for it, but there's too many stories coming out of players and too much influence, and I think that should be stopped, and there should be a greater level of independence, and they should only issue advice. You can't be in every country telling every country what to use. Especially we wrote a manuscript on the COVID policies and stuff, and some of them were obviously wrong. So if these policies were mandated like strictly around the world, wrong policies, I mean, that's definitely not right. If they got the science wrong and then they're saying, but you must do that. No. I mean, that's what I like about the old people. They say, oh, well, maybe we done this or maybe we done that. And you can do that if you want, I don't want any police chasing me along the beach because I'm just jogging for some public health. And then that's what happened, So they've gone too far. They should be drawn away back and maybe abolish it and reconstitute something, Share knowledge for, I mean, we have the internet, share knowledge and coordinate knowledge, but no, I don't know if there's a trough of food or money, all the picks just descend and, it's not good. Human beings will be greedy and lust after power and money and all of that stuff. And that's what's happening just now. I don't, some of the motivation, I'm like, well, have you not got enough money? Do you need actually more, or power, or no, it's corrupted. That's the old fashioned word. It's been corrupted, and I don't even know if it should be torn down and called something else, rebuilt. And, we need people that, really, when I write a paper, they say, what's your conflicting interests? Okay, they don't do that for the WHO. They have a million, Really, we should really, really get back to that, and for scientific journals, obviously, because they are conflicted as well. So
Okay.
that's it, But you can send me any more questions by email, Tom.
All right. Yeah, I've really enjoyed this conversation. Do you have any other points you want to make before we finish?
Do I have any points? No. Just I'm really interested on all of these different. I think back to your AI point. This combination therapies. There are experiments on some of the other molecules. I mean, I think we should be able to go faster. This idea has been around for about 10 or 20 years. We should be able to go faster with genetic sequencing and AI on the. Can we formulate a new drug response rather than just antibiotic? Is there a better process, a more sustainable process, and a healthier process? And thanks very much for having me on, Tom.
Oh, no problem. I really enjoyed it. I have to have you on again. Dr. Gerry Quinn, thank you.